Hello, Namaste. I am Varsha Gaitonde and welcome to my channel. In this video, I am going to tell you about the physiological basis of heterosis. There are different mechanisms in order to support the physiological basis of heterosis. The first and foremost is net assimilation rate. If there is increased net assimilation rate, heterosis increases due to increased availability of nitrogen to the roots. As you know, the hybrids are the voracious feeders of nitrogen. They exhaust all the nutrients from the land, or we can say from soil, and by this process they increase the net assimilation rate, due to which the vigor of a plant is increased. The second is leaf area index. Heterosis helps in increasing both sink and source capacity. High leaf area index as comparing to their parents gives better survivability and better productivity as comparing to their parents. The third thing is high root growth. Roots help as a better sink for photosynthesis. Then hormonal balance. If you can say there is some hormonal good amount in one parent and some hormonal composition in other parent, when by their combination we can make a favorable balance in the hybrids. As I already told in other videos also, whenever you are going for heterosis or going for hybrids, all the hybrids which you have developed cannot give you the best one. The only hybrids which are best as you can obtain are the hybrids which are having better combination and compatibility with each other. If you are developing thousands of hybrids, one or two hybrids which are very favorable, which are giving good performance in multi-location trial and multi-year trials, can be released and grown in the further generations and can be utilized for the commercial exploitation. Hormonal balance. Better balance gives superior performance than their parents. Example, hybrids responds to better endogenous gibberellic acid concentration. Then metabolic reaction. Inbreds have elevated metabolic reaction, whereas their combination or hybrids may give good metabolic reaction. Mitochondrial complementation. Based on the in vitro studies, somatic hybrids and hybrids which have better complementation, as some of the plants may be having little or less effective mitochondria, but some others are having good mitochondria amount. Then by combining these two, we can get the better complementation in the mitochondria. As you know, these are involved in the energy induction in the cells. Next is the causes of heterosis in terms of physiological aspects. First one is greater initial capacity hypothesis or greater initial capital hypothesis. The theory was given by Ashby in 1930. He studied the physiology of inbreds and hybrids of maize and tomato, conducted that hybrid vigor is due to an increased initial embryo size. He termed it as greater initial capital. I just would like to share the Ashby's hypothesis of greater initial capacity. Here, heterozygosity results from the greater initial weight of the embryo, and hybrid vigor is nothing more than the maintenance of initial advantage in hybrid size. Hybrid do not differ from their parents in relative growth rate. Ashby's hypothesis is explained in terms of three things. One is greater initial weight, followed by maintenance of embryo size in the initial stage, and relative growth rate. This you need to remember. The second cause is cytoplasmic nuclear interaction. Michelis, Shell, Lewis and others suggested that hybrid vigor is the interaction of cytoplasmic and nuclear systems. Cytoplasm is a transparent fluid rich in RNA and mitochondria, which is usually transmitted through the female parent to the offsprings. Coming to the stages of physiological manifestation of heterosis. The first stage is seed and embryo development. High positive correlation was recorded between hybrid vigor and embryo weight. Copeland noted that hybrid embryo exhibited greater vigor in the earlier stage of development. Ashby found significant differences between embryo weight of reciprocal crosses. Early difference in the embryo size in favor of hybrids resulted from a rapid growth rate of multicellular zygote. That means there would be better compatibility if we go for hybrids, and this will show the initial vigor as well as initial higher embryo development due to the complementation of different physiological aspects. Second one is early seedling growth. Hybrids show higher growth rate than the inbreds immediately after germination but not later. They do not have a larger maximum growth rate period. Ashby say embryo size is more significant than reserve food material. As you can say, hybrids can complete their life cycle, means in order to say, if we talk about the reproductive stage, they try to complete in a better way as well as in less time. So it's a beneficial aspect related to hybrids. Third is later seedling growth. Hofer found root dry matter content of hybrids to be intermediate between parents. Hybrids found to absorb and utilize more amount of NPK than the inbred lines. Catalase activity was higher in heterotic hybrids. That means enzymatic activity, biochemical molecular complementation as well as the better nutrient absorption capacity is found in the hybrids in later seedling growth as well. Thank you. I hope this information is helpful for you. If you like this video, do not forget to hit this like button and please keep subscribing, sharing and commenting. Thanks once again.